Swayam Prabha. Digital India. Educated India.
Good morning, everyone. Anything on biology you heard? What was the topic on biology? Molecular? Okay. And then any other biology subjects? Biophysics. Instrumentation, is it? Biophysics means molecular modeling, structures? No. Pardon? Electrophysiology. Okay. That's good. So you are all doing projects? All of you are doing projects actually. Anybody in biology projects? Okay. I'm going to talk two different topics. Morning, first hour, I'm going to talk about biomaterials, medical biomaterials. And then later on, we'll talk about polymers and so on. So have you all gone to dentist before? Any of you have gone to dentist? Yes. So what did the dentist do? Oh, a lot of you. You must brush your teeth better. So what did you do there? What did they do? Did they clean? They put in something inside? They put some material inside, right? No? Anybody had any material put inside? Yeah. So teeth, they must have put artificial teeth, is it? They removed your teeth. They put in something else? They didn't put. Okay. Anybody had any material put inside? Yeah, you had. Yeah, amalgam. Okay, so you had a implant placed inside, right? Dental is one area where they put in a lot of material inside, right? Sometimes you have artificial tooth. Sometimes they put in amalgam. If there is a hole in the teeth, they put amalgam. Artificial teeth is made up of ceramics and so on. Okay, so many different materials is put inside the teeth actually. Polymers are put inside, metals, materials, and so on. Did anybody have anybody of your relatives had fractures, bone fractures? Yes, you had some relative. You yourself. So did they put any material inside, metals? No. Okay. So if there is a big, when there is a big crack, big gap in the bones, they put something plates, right? So stainless steel, titanium, that sort of materials are put inside actually. Okay. Contact lenses. Anybody having contact lenses? Nobody. So if one of your relatives has contact lenses, you can touch it and feel it. It will be very soft. Okay, that's called hydrogel. It's a soft contact lens. It's made up of polyethylene glycol. So contact lenses should be. What are the properties a contact lens should have? Do you think?
It shouldn't irritate the eye. It should be transparent.
Yes, very good. It should not allow bacteria to settle down. It should also allow oxygen to flow through. So if the oxygen doesn't flow, your eyes can become red. So those are properties of those contact lenses. Now somebody had plates. What should be the properties of the plates? Yes, non-corrosive. It should long last, very long, right? And then what else can you think? Yes, it should not create bacterial infections. Okay, so that's, have you heard of angioplasty? So what do they do in angioplasty? Anybody heard of angioplasty? Yeah, very good. They put in a stent, right? Because the arteries is compressed. The blood doesn't flow. Why does the arteries get compressed? Because of plaque formation, a lot of fat deposits. So it's made up of titanium nickel. It's called nickel titanium alloy. So what should be its properties? Yeah, it should retain because it can get compressed more. And what else? It should not activate the platelets and the red blood corpuscles. Once those get activated, you can have thrombosis. You have heard of thrombosis? That means accumulation of the red blood corpuscles and it forms the thrombus, it's called. So it should have a different properties. So biomaterials which are kept inside the body should have different properties depending upon where it has to be there. Stents should last forever, correct? The person who has, you have seen somebody getting a blood transfusion, right? Or glucose transfusion. So those are also biomaterials, but it stays only for a few hours. So some biomaterials for a few hours, some biomaterials, for example, the blown bone plates have to stay for a very long time. Stents have to stay forever. You can't replace stents regularly, right? So these are materials which are put inside the body, and it should last either for short period or long period, and it's in different places of the body. If it is orthopedic, then it could be in the leg, near the bones. If it's a stent, it's near the blood region, right? If it is a contact lens, it's near the eye. So it's in different parts of the body. It stays for different time periods also, okay? So a biomaterial, if you look at the definition of a biomaterial, okay, a material, it could be synthetic or natural, okay? I will show you what is synthetic, what is natural, okay? Used for some time as part, or sometimes it replaces also. It can be completely replacing the part, or it could be as part of the body. To treat, augment, diagnose, replace tissues, organs and functions of the body for a short period or for a long period. If you are talking about, you have heard of urinary bags. What is it used for? So if somebody is having an operation and on the bed, they can't pass urine, so urine automatically goes and collects in a bag. So that lasts for a few hours, then they remove that, okay? Stents last forever. Your stainless steel plates or titanium plates in the bone fracture again last forever. So it could be short duration, long duration. It sometimes augments the function, sometimes it completely replaces. There is something called left ventricle assisted device. You have heard of that? No? Anybody? So sometimes the heart, the left side of the heart, left side is more important than the right side of the heart, right? Why is that? Because it pumps blood to all parts, right? So it should be much stronger, like a pump, much more stronger. So some people have very weak left side. So the blood is not getting pumped enough. Okay, it could be by birth or it could be because of some health reasons. So there is a mechanical pump which is put, that's called left ventricle assisted device, to help the left side of the ventricle to pump better. Okay, so that's also placed inside for a very, very long time, because those patients cannot have enough blood pumped throughout the body without that particular assisted device. So it is assisting the existing heart. Okay, so it's of assistance. Sometimes you can think about replacing also. Somebody having a cataract surgery, right? You've heard of cataract surgery. So they remove some parts and then they put in artificial inside. So that is completely replacing. So there could be situations of that nature also, actually. So it could be assisting the existing part, or it could be completely replacing the part or the organ also, actually. Okay? So that's called a biomaterial. So two important things are there when we design biomaterials: the biocompatibility, because it's going inside the body, right? So it should be biocompatible. It should be compatible to the human the person who is going to have it. Otherwise, it should not be toxic. It should not create problems, the long-term effects, short-term effects, and so on. Actually, the biocompatible. It should be compatible with the person who is going to have it, short period, long period, and so on. Actually, okay. And how the body reacts to that, because this is a foreign material you are placing inside. So how the body will start reacting? As you know, our defense system is so fantastic. Anything that's foreign, immediately they start attacking. They will have enzymes which will come and attack. They will have T cells, they will have immune cells. They all come and start attacking the material you are placing inside. So how the host reacts to that material, that's also very, very important, actually. So how the material is affecting the body, how the host reacts to the body. So when we design biomaterials, we need to consider all these aspects. That's very, very important, because like we talked, some materials are placed in the bone region, some are placed in the heart region, some are placed in the eye region. So different locations, different functions. So the material has to survive the host response also. Okay, I just want to show a lot of pictures actually. So this is called a stainless steel bone plate. So if there is, these are X-rays. So if the bone breaks and there is a gap, so they keep these stainless steel plates, and these holes are meant for screwing. So they screw these plates to the bone. Okay. These are made up of stainless steels because stainless steel is cheaper. But the problem with stainless steel is it's corrosive. It sort of gets corroded because stainless steel has iron. Okay, so that's the problem. So there could be corrosion of that, but it is cheaper. Stress. Somebody said stress concentration. You said no. That will come with any metal. I'll talk about that. Yes, it's called. So the problem is stainless steel. It's cheaper, but it slowly can corrode also because it contains iron. And somebody who has diabetes, then the corrosion is much higher. So that's a problem with stainless steel. So they started going with titanium. These are titanium plates. Titanium is expensive, but in India we are still, the cost plays a very important role. So we still use stainless steel quite a lot in India. So these are titanium. Titanium doesn't corrode. You can see some of these titanium plates. The holes are meant to screw the plates to the bones actually. You can see the X-ray pictures how they are screwed here. Sometimes they use metallic screws. Sometimes they use some polymeric screws also. The polymeric screws can slowly get degraded over a period of time. If you have metallic screws, it will remain forever and ever actually. So you can see those screws where the plate is screwed on to the bone here. These are titanium screws. Okay, these are similar stainless steel plates. What happens is sometimes there is bacterial contamination because there is always bacteria inside the body. When there is an open wound, bacteria gets inside, infection happens, right? When you have a cut or when you fall down and have a big wound, you always get infection. Doctor gives you antibiotics. The idea is to kill those bacteria. So these bacteria enters and they circulate inside the body, mostly because of wound. When we have accident, when we fall, when we have a cut and so on. So these bacteria can go on accumulate on these biomaterials. That's another problem. Whenever any material is placed inside the body, bacterial contamination happens and the bacteria starts growing inside. Okay, so we have to, doctor generally gives antibiotics when they do this type of surgery. Any surgery, doctor always gives antibiotics. That's called systemic antibiotics because it's going through oral and it's going and killing the bacteria. But the place where the bacteria is growing is near the plates where you have implanted. Okay, so what we do is we try to coat the plate with a polymer called the polylactic acid with some drug. So the polylactic acid slowly degrades and then it slowly releases the drug. So that if bacteria nearby the plate will get cleared. Okay, that is more like a local delivery. So you have two things. One is systemic delivery is what the doctor tells you to take antibiotics 2 times a day, 3 times a day. We also have local delivery. What we do is we coat the plates with a polymer called poly-L-lactic acid, which is degradable, which also we put in some drug there. So when the lactic acid, polylactic acid slowly degrades, drug gets released. So bacteria which is very close by will get killed actually. That's a local delivery. So we use a lot of polymers which are biodegradable. There are polylactic acids are there, chitosan is there, polylactic glycolic acid, polyethylene glycol, like that. There are many polymers which degrades inside the body. So when you mix it with drug and you have a coating and the polymer degrades, drug also gets released. So nearby region will be full of antibiotics. So if there is any bacteria, they get killed actually. So that's called a local delivery. So what we do is we coat it with this particular polymer in this case we have used silver. As you know, silver is an antibiotic, right? Once those days silver nitrate they used to put if there is a wound or cut or burn and so on, because it's got antibacterial properties. So one of the big problems with in plates is bacterial contamination. Bacteria can go and sit and then slowly got spread and then the plate will be full of infection with bacteria, then which can lead to inflammation, pain, and so on. So that's a very important problem in any implant. When we keep an implant inside, that problem is always there actually. So this is called polylactic acid, which is biodegradable. So you can have some of these screws made up of polylactic acid. Okay, the screws could be polylactic acid, which will after a period of time slowly degrade. So the screw will completely disappear, only the plate will be there with the bone actually. Okay, so this polylactic acid is a biodegradable polymer. So they make screws out of it. And these screws are useful when there is a ligament tear. You must have heard of ligament tear. Sportsmen who run as sprints, they always have a ligament tear. So the ligaments are connected with these screws, which are made up of polylactic acid, which is biodegradable. So over a period of time, it will completely disappear, and the ligaments will get healed actually. So that's one application of these biodegradable polymer. This is a polymer called polymethyl methacrylate, PMMA, polymethyl methacrylate. It is also used for delivering drugs. So if there is an open wound, like I said, when there is an open wound, say after some accident or trauma, a lot of bacteria comes in, and then the whole place gets infected. They not only give antibiotics, but also local antibiotic delivery. So what they do is they fill these beads with some antibiotic, and then they keep it inside and stitch it. So the drug slowly gets released from those beads. And they use very strong antibiotics like vancomycin, like meropenem, and so on actually. So these polymers are not biodegradable. So the problem is once all the drug goes away, the polymer will be sitting inside. So we have to they make a small hole and remove this particular polymer. So they are not biodegradable. So ideally, if you have some biodegradable polymer with drug inside, drug can get released, and after that, polymer also can slowly degrade. But currently, they use this polymer, polymethyl methacrylate, which is not biodegradable. These methacrylates are also used in many places actually. Does anybody have idea? Methyl methacrylate, polymethyl methacrylate, and so on. Especially in dental, it's used. The teeth, artificial teeth. Now they make ceramic-based tooth, but before that they used to make polymethyl methacrylate tooth. Acrylate cements, when they want to connect some broken tooth with metal, they use cements. They're all made up of methacrylate actually. Even doctor, if you have seen, he fills it up and then puts in a light inside, and then the whole thing gets fused actually. So they use this type of methacrylate type of polymers. But these are not biodegradable. That's why they are used in dental. But if you're looking at these type of application, we want the drug to get released, but later on the polymer also should completely disappear or degrade, but it doesn't happen. So there is a lot of research opportunity for doing research in designing new drug delivery systems which will deliver the drug and they also will slowly disappear or degrade. Okay, that's called biodegradation, or even that's called bioresorption. Bioresorption. So the entire polymer will get resorbed in with the body fluids. That's called bioresorption. Whereas in biodegradation, the polymer will degrade into small molecular weight, and it will come out through the various fluids outside. Okay. Whereas in bioresorption, the entire polymer will get completely absorbed. Okay. So there is a difference between bioresorption and degradation. So degradation, polymer, big molecular weight will get smaller, and then they will come out of the body through the body fluids. That's called biodegradation. So these, ideally we should have some polymer which will deliver the drug, and then slowly it gets completely bioresorbed. That means completely it will disappear. So that will be nice to have actually. Okay, dental, lot of materials are used in dental actually. Lot of materials, polymers, metals, and ceramics and so on. Root canal treatment. You have heard of root canal treatment? Anybody had root canal? You had. Okay, so the tooth is removed and then they'll make a hole and then they will fill up with some antibiotics so that all the infection goes away. They will ask you to come 1 week later and then they will put in a metal like this, you know. This is called a metal post. That's a metal post. It's made up of titanium. And then the tooth is put on top of it, right? So you have the tooth on top of this metal post. So this material is made up of titanium. You can see the next picture where it is fixed nicely. And then later on, they'll put an artificial tooth and they will fuse it to that actually with an adhesive. Okay. So anybody having root canal treatment, they will have this titanium, then they will have an artificial tooth which will be put actually. These are called inlays. These are made up of ceramics. So if there is a big gap in the tooth because of any reason, why the big gap come, either broken or if there is an infection, bacteria, infection, then they remove the bacteria, so there will be a big hole. Okay. So they put in this type of material and then fix it, okay, with an adhesive. These are called inlays. These are impression materials. So if somebody, if they want to make an artificial tooth, they'll put an inlay, this type of impression material and collect the impression of the tooth region. More of this. So you can see metal crown. So the tooth there is completely damaged. So they put in these crowns, metal crowns. Different types of metals are used. Sometimes ceramics are also used nowadays. A lot of interest in ceramics. Metal, stainless steel type of material, but in the front you don't like to have stainless steel because it don't look nice, but in the back maybe, so they are called metal crowns. So the top portion of the tooth is covered with these type of material. So inside it's also damaged, the tooth is damaged actually. Of course, braces, many of you maybe might have had these braces, right? Metallic braces are there, now even polymeric braces are there which will match the color, so that you will not know whether you have a brace at all. So those are polymeric braces. These are some, see there is a big gap because of infection, okay. What do they call in dental? Anybody knows? If there is infection, bacteria, they call it caries actually, caries. So that's because of the bacterial infection. So they clean it. That means they remove all the bacteria, dead bacteria, everything. So there will be a big, almost a hole like will happen. So they will try to fill it up with some material. It's a polymeric material. Then there is an adhesive added, actually 2-3 polymers added. So methacrylate will be there. And then it gets completely filled actually. So these are called composite fillings. They last for many years. Unless you eat bones or something, they will last for a very long time actually. Amalgam. Mercury, different types of materials are used. That is, if there is a small hole, they'll just use mercury type of amalgam to close the holes actually. Why do they use mercury? Any idea? It's a bit soft, so it nicely fills up the gap, right? So that is why mercury is used. Yeah, so that it was used and slowly they are coming out of mercury. But still it is used because there is a lot of studies which is being done that mercury is not getting released, and many studies are there, but they do want to come out of mercury actually. A lot of, like I showed metal crowns, these are ceramic crowns, because nowadays everybody wants the aesthetic very important, right? Nobody wants metals. Once upon a time they used to have stainless steels. You have seen in an old James Bond movie, The Spy Who Loved Me or something. You have seen? Anybody knows James Bond movies? You have heard of James Bond movies, right? Okay, you heard. There was a villain, and villain had one assistant, big guy, and he had metal teeth, stainless steel. So he can bite anybody with that stainless steel teeth actually. He can even bite some metal locks and things like that. So, but then nowadays nobody likes metals because aesthetic is very important. So a lot of ceramics are coming into market because it will exactly look like your tooth. You will not be able to differentiate. Otherwise, people had metal teeth, gold teeth was there, silver was there, stainless steel was there. So nowadays everybody wants it aesthetic. So these are all becoming more to match the color of the person, tooth of the person. In fact, they have different colors so that they will compare your tooth with those samples, and then they'll use the corresponding tooth actually. Veneer. Okay, if you have seen nowadays movies or TV shows and all that, now everybody smiles and they have beautiful tooth, right? Beautiful teeth. Definitely it's not true, because they must be having very lousy teeth also. But they stick these nice looking things which are called veneer. So they can, with an adhesive, they stick to the front. So the entire portion will look very, very nice actually. These last for about 3 years, 3 or 4 years, depending upon how you maintain. So if you see all the movie actresses or even television show actresses, when they smile, they will have nice looking teeth. But that's not true at all, because these type of cosmetic things are done. These are called veneer. So they stick it, as you can see in the top portion. That's how the teeth may be for a person. And once they stick this, it will look so nice. Same here actually. And it can match exactly the color of the person, the color of the teeth of the person. No, they have different hues will be there, and they will select the correct one and it will exactly match. So individually it is stuck. Each one is stuck individually. So you can have veneer stuck to all of them, and you get beautiful looking teeth, which you can use it in your TV as an actress. These are called veneer, and these are made up of ceramics. You can match exactly your rest of the teeth color. That's the beauty of it actually. So basically they will just stick it with an adhesive to the existing tooth. There are always problems out there. Sometimes the place where they had implanted can get inflamed. There could be inflammation because the host is reacting to the material. So that's a host related reaction. So you can see the inflammation for the patient. They may have to remove that, then cure it. That means they have to give drugs, anti-inflammatory drugs and so on. And then they may again replace with another material. So the material has affected the host. So the host is having a reaction to that material. Sometimes there are people who have toxic reactions towards nickel, towards chromium, and so on actually. So maybe the person is getting affected because of those materials actually. So that can happen in many cases actually. In metallic implants, some may not have any effect on these type of materials. Some may get affected on these type of materials. So it's not uniformly same. So that's another challenge when you design biomaterial, especially when you're using metals which will have nickel, chromium, and cobalt, and so on. So those problems could be there. And another thing is bacteria can sit on it. These are some confocal laser pictures where the bacteria is sitting on this material and they start spreading and so on actually. So that is possibly another problem with these materials. So this is how the host seem to react to the material, and this is how the bacteria seem to react to the material which is placed inside the body actually. So in all these biomaterial, this problem is there, how the host reacts. So one person may react differently, another person may react differently. If you are having a plate placed after an orthopedic surgery, some people will not have any problem, but some may have problem. Same thing can happen in dental also. Some will not have any problem after this dental implant, but some may have problem because of the material. Different types of materials are placed in the dental area, like I am showing actually. And these are called urethral stent. So these stents are placed, as you know, from kidney, the urine flows to the bladder, right? There is a small tube which is called ureter. So the urine flows through that small tube which is going to the bladder. Sometimes this tube gets blocked because of stones. People get stones, right? You must have heard about stones. Or sometimes inflammation, infection. So the tube gets blocked. So urine doesn't flow coming down to the bladder. So they place something called urethral stent. Okay, it looks like this. This looks like a long. It's made up of polyurethane, almost like rubber. It's quite flexible actually. It is placed in the ureter region. As you can see, this is the bladder. This is the bladder, and then this is the kidney. there is a ureter, this is X-ray picture. So the stent is placed. Okay, urine is a very, very harsh environment. You have bacteria, E. coli, protease, those type of bacteria are there. And then urine also contains a lot of salts, right? Because people get stones. You must have heard about stones, people having stones in and so on actually. So urine is a very harsh environment. So when you keep this type of material inside, you have a lot of bacterial contamination happening, bacterial settling, salts settling. So all those problems are there when you are designing material like this. So these are called ureteral stents made up of polyurethane. As you can see here, it is in a very, very, very harsh environment in the where there is urine flowing actually. So when you have cardiac stents in the heart region, you don't have these type of material, but you will see blood there. Okay, so different environments, different types of materials we are talking about actually. These are called knee replacement. Have you heard of knee replacement? There are nowadays a lot of people are getting the entire knee replaced because overweight, that is obesity, because of certain diseases where the knee joint gets fused. You have a ball and socket which moves, right? The knee gets moved. So the whole thing gets fused. They can't bend the knees also. So the entire knee is replaced. As you can see here, the entire knee is replaced. So it's got a metal crown, and there is a metal ball which is going round and round, and there is a polymer inside, which will be like a lubricant actually. So in knee replacement, we have nickel, chromium, ball and crown, and polyethylene as a spacer so that there is no metal on metal contact actually. So knee replacement surgeries also, they use these type of material. And this is near the heart region. You have the heart diaphragm valve, okay, heart valve, and they use titanium, cobalt, alloy, and then polyester, polytetrafluoroethylene, some polymers, polyesters and polytetrafluoroethylenes are used actually. Okay, so this is called a valve which opens and closes in the ventricle region for allowing the blood and in one direction, right? So these are again made up of metals and polymers actually. So have you heard of Sri Chitra Tirunal? Anybody has heard of Sri Chitra Tirunal? It's in Trivandrum. Maybe those who are coming from Kerala may know. Sri Chitra Tirunal Institute of Higher Medical Sciences. Anybody heard? Those from Kerala must know. No? It's in Trivandrum. It's one of the best biomedical institutes where they design a lot of biomaterials and they help to market a lot of biomaterials. Okay, so Sri Chitra Tirunal Institute of Medical Sciences. So they also made these type of heart valves. So if you import the heart valve, it will cost about 2.5 to 3 lakhs, one valve. But Sri Chitra Tirunal Institute of Medical, they have a valve which is about 60,000 rupees. They have reduced the cost by almost 3 times. So it's very, very affordable for Indians. So they do a lot of research in biomaterials and help in marketing like blood bags, urinary bags, valves, these type of valves, and so many, many items actually. Sri Chitra Tirunal Institute of Medical Sciences, that's in Trivandrum, Kerala. So these heart valves, you can see so many materials are used in making the heart valves. No, titanium, cobalt, then polymer like polyesters, polypropylene, and so on. So sometimes some biomaterials they use very complex type of list of materials. Sometimes it's just one material. For example, if you look at this chap, this is only polyurethane alone. Whereas if you look at knee replacement, there are metals and polymers. And here also 2 metals and many polymers are used also. Okay, so this is a very complex material which is placed inside the heart. So it allows the blood to flow in one direction, does not allow the blood to flow in another direction. So these are very, very, very precision based material. These are something called shunt. This is the white thing is made up of a polymer called polytetrafluoroethylene polymer. You have heard of polytetrafluoroethylene? We use it in kitchen also. Yeah, Teflon we call it, where the pans they coat it. Why is it coated? It's very smooth and non-sticky, right? So this polymer is polytetrafluoroethylene, and it's very inert type of material, right? So this is used in these type of these are called shunts. Okay, these shunts are used to connect aortic branch and pulmonary artery. Aortic branch, you have heard of it, you must have studied in your biology. Yeah, what does it do? The blood from the aortic branch goes to the body. Only the pulmonary artery takes the blood to the lungs to get oxygenated. Okay, so what happens is there are some babies who are born with a hole in the heart. Okay, so the blood does not go to the lungs to get oxygenated. So those babies are called, you heard of blue baby syndrome. So such babies, because they have a hole in the heart, the blood does not go to the through the pulmonary into the lungs to get oxygenated. So oxygenated blood is, what's the color of oxygenated blood? Red. Unoxygenated is blue. So such babies will look little blue. That's why they called it blue baby syndrome. So these shunts are placed here. So the blood from the pulmonary artery, the blood from the aortic branch is taken to the pulmonary artery to the lungs actually, so that they get oxygenated. So it's called a blue baby syndrome actually. So these are made up of PTFE. So the babies are born with a hole in the heart basically. So normally they have to do a open heart surgery to close the hole, but the babies are so young they can't do a open heart surgery. So
Sir, isn't Teflon carcinogenic?
no, it's not. It's very inert actually. It's not carcinogenic. We are talking about room temperatures. If you are talking about your pan and all, 240 dry, you can have some toxic chemicals released. Here we are talking room temperature 37 degrees inside the body. It's a very inert material, so it will not activate the red blood. That's the main idea actually. It's used. So these babies are born with that. So they can't do a open heart surgery to close the hole. So for few years until they become older, they keep these type of shunts. Once they become older, 10 years or 11 years, then they will do a open heart surgery and close the holes actually. Okay. So generally these are placed when the baby is 2 years old, 2.5 years, 3 years, in that range actually. So they are born with that defect. That's called the blue baby syndrome. So these are polymer polytetrafluoroethylene. That's an angiogram of that actually. So what is angiogram? You have heard of angiogram? They pass a dye. Then they take the CT. So that it's a color of the dye, which is that generally about blue color. Then for visibility, so they inject a dye. Okay.
Sir, you said that people who are born, babies that are born with these small holes in their heart, will get the surgery done only much later.
The open heart surgery done later, but they do this particular shunt very immediately.
How will they do that without opening the heart?
They will, because the baby will otherwise die.
So how will they do it?
How will they do? Means they will just open this portion where the heart is, and then they will connect this particular shunt, and they will suture it here, and they will suture it there. So they will outside. They will not open the heart. If there is a hole, as you know, we have the ventricles, then you have this separation. So there is a hole. So they have to open the actual heart and then close the hole. So they are not opening the heart. They are just connecting with this tube. So that is called open heart surgery. You have to actually open the heart. You have the ventricle. You have the separating. You have 4 compartments, no? So there they have to do that.
Sir, isn't the blood in the aortic branch already oxygenated blood which comes from the ventricle?
No, see, when there is a hole, there is a short circuit. So you have 4 compartments, there is a hole. So the blood, instead of going to the pulmonary region in the right side, some of the blood will come into the left side. So that we want again oxygenate.
So it's not completely oxygenated?
Not completely.
Okay.
maybe partially. So suppose if there is a hole and you have to pump it, more can come through the hole, escape. So many 80% is not oxygenated actually, hardly 10 to 15%.
Sir, what does BT stand for?
Okay, BT are the names of the people who invented this. Originally, they tried rubber tubes. They tried different tube material. They even took from the calf their veins and then stitched it. Finally, in the past 10 years to 15 years, it's now polytetrafluoroethylene, because these are recent polymers. Otherwise, they tried animal veins like that, rubbers and so on. Actually, see, these are two people who actually worked on it and invented this. There is you have heard of HBO. HBO you have heard of, right? They have a lot of movies there. There is a movie called the baby the God did, or something like that. If you Google it, I don't know whether you can see the movie fully. You may have to pay it. So that talks about how they make it like a movie with a little bit of exaggeration and all that. So the people who worked on it and how they came up on this idea and so on. Actually, it's called the baby. So there are 2-3 people, one pediatric surgeon, one normal surgeons. They tried different materials, and finally initially they tried with the rubber and it worked. This baby completely got cured, the blue color changed and became. Then they started doing R&D on the material, and then finally we have this PTFE. It's a polymer. I'll give you the name of those 2 people. Actually, I don't remember the name of the movie also. It's a HBO movie. So it's not a regular movie, but it's HBO. Okay, so one of the biggest problem is this bacteria settling on the material. Whenever we keep inside a liquid and solid, bacteria start settling. If you have seen your home buckets, right? You have water after a day. If you feel inside, you have felt it. Have you felt inside a bucket in the bathroom, for example, when there is water? Anybody felt that? Water which has stayed inside the bucket for a day or something? Nobody felt that? You all have baths, no? Correct? So we have bucket, we have water flowing inside. Unless if it is shower, then you will not feel. But if you do it with the bucket, there will be already some water inside, plastic bucket. Have you all felt inside? Anybody felt? Yeah, how does it feel? Slimy, that's bacteria. Immediately within a few hours, bacteria will come. So this bacteria produces something called exopolysaccharide, EPS. So that's the slime part of it. So the bacteria produces slime, which is very slimy like. That's called EPS, exopolysaccharide. So you can see, and the exopolysaccharide acts as a protective layer for the bacteria. So any material and liquid, immediately bacteria will start settling down. It will produce this EPS, and then it will cover, because the EPS acts as a protective layer for this organism. So you can see different types of organism on polyurethane producing. These are some scanning electron microscopic picture. This is called Pseudomonas aeruginosa, and this organism is found more in the, anybody? This particular organism, Pseudomonas aeruginosa, is found more in the, it's involved quite a lot in different types of pulmonary diseases. So if you have cough, cold, cough, you will find this Pseudomonas aeruginosa actually. Animals, they also spread this because it comes through their sputum and all that. This organism, Staphylococcus aureus, and this is very ubiquitous. It's found quite a lot, even in our skin and all, it's found. But only if there is an injury, it can go inside. Otherwise, our skin will always have Staphylococcus aureus. So that's quite infectious, the Pseudomonas aeruginosa, because it's involved quite a lot in the pulmonary region. So if somebody has lung infection, if they take out samples, you will see that sort of bacteria actually. So these bacteria, as I said, once you have a biomaterial placed inside, immediately they start spreading and settling down, they produce EPS and they'll live happily there actually, right? So we need to think of ways of preventing that. Any biomaterial, we place it inside, we need to think of this problem can start within a few hours, and we are talking about biomaterials which are going to be there for a very long time. So they have to think about how to prevent that. These are called hydrogels. This is a contact lens. If somebody has in your house, take it out and feel it. Okay, if they allow you to take it out and feel it, it will be very soft and pressable actually. You know? These are called hydrogels. It's made up of a polymer called polyethylene glycol. It's very soft and it will take a lot of water inside. It will always be wet. So it is used in wound healing, wound dressing quite a lot, because wound dressing should not get dry. So they have this type of, these are called hydrogels. They take in a lot of water, absorb a lot of water, and swell. There are also called nowadays super absorbing polymer. Okay, they are used in diapers, because diapers, urine, lot of urine comes in. They should be able to take in all the urine and remain almost in the same size actually. You know? So there are super adsorbent polymers are there, which does this job. But these hydrogels don't take up so much water. They take up about 4 times, 5 times. But super adsorbent polymers used in diapers can take up almost 10 times the water. So 1 gram can take in 10 grams. Okay. So those are used in diapers. These are mostly used in wound healing patches. These are used in your contact lenses and so on. So these polymers, these are polymethyl methacrylate. Like beginning I said, you can put in drugs inside, so the drug gets released. So we can use it as a drug delivery, but they do not degrade. So they remain after it's given up the drug. So doctors do another surgery to remove it. So now they are thinking about other biodegradable polymers. So if there is a wound, for example, you can see a stainless steel rod here, right? And then they also keep this beads with a drug called meropenem, and then they close. So this drug gets released and it kills all the bacteria nearby. Later on, they have to remove this because it does not biodegrade actually. So especially when there is an accident, big wound, so a lot of bacterial infection. So if you can design polymers which will also slowly degrade, they don't have to do a second surgery. So that's an advantage of this actually. So you can see there are many biodegradable polymers. Polylactic acid, polylactic glycolic acid, these are all biodegradable. You can see now in 30 days how they degrade. So we can design different polymers where some may degrade if you want 6 days, some if you want 30 days, some if you want 45 days. So whatever inside the polymer, drugs can slowly come out. These are some scanning electron pictures. So we can have drug delivery polymers which will degrade depending upon the time for which you want it to design actually. Like I said, 1 week, 30 days, 45 days also we can do, and slowly it can release the drug, antibiotic for example, so that bacteria nearby gets killed. Okay, or we can, yeah, go ahead.
Sir, you told these polymers like after they deliver the medicine they will degrade, but like
No, not after.
like
They will slowly
whatever
degrade
time we
and
want.
drug also will slowly come out.
So, even if they're biodegradable, the compounds which will come out after degradation, how will they be removed? Like they might not be very good for our body, or they might be even toxic for us.
Yeah, okay. We check for toxicity. All these are approved by FDA. FDA is the Food and Drug Administration of US. So they do the approval. They look at the toxicity of this material. They look at the toxicity of the degraded products also. And then they also look at the elimination from the body. If it is biodegradation, like I said, it will become small molecular weight and gets eliminated from the body. So they look at the elimination of it also over a period of time. Okay. So they do all those tests. We need to do all those tests. Toxicity of the material, toxicity of the degraded product, how it gets eliminated from the body.
Sir,
The degraded
how
product.
do they get eliminated?
Pardon?
Will they go to the, like, will they be filtered out by the body itself?
Yeah, they will get
Or
eliminated.
is there any process which should be conducted from outside?
It will get eliminated from the body. There is something called pharmacokinetics. When you take a drug, okay, so drug acts, suppose I take for fever, it goes inside and it acts. And there will be many side products, it has to get eliminated. How does it get eliminated? What are the routes? One route is your urine, another route the fecus, another route is through the exhalation. So in animal studies, what they do is, that's called preclinical studies. Before human studies, they do animal studies. So they will give the drug and they will monitor how much is coming out through the fecus, how much is coming out through the urine, how much is exhaled, and then they will do calculations. So they will find out that actually. So these are the elimination routes. Only 3 routes are there: the fecus, the urine, and the exhalation.
Sir, can it come out of the sweat or sweat?
Sweat, I don't think rats don't sweat.
No.
Mostly those things are done on rats, so they don't sweat. See, what happens is, I have to go deeper. You have the liver. Liver does a lot of biotransformation of any object that comes inside. Okay, biotransformation. So in the biotransformation, liver is a very good gatekeeper, because whatever foreign material comes in, whatever foreign material or xenobiotics. So what liver does is, they biotransform. They will add oxygen or OH groups to the molecule using enzymes. So once you add OH or nitrogen group or oxygen group, what happens to the molecule? It becomes more, you have heard of hydrophilic? Hydrophilic, right? It becomes more water soluble. Okay, so liver does that. There are enzymes called oxidoreductase, which adds oxygen to whatever xenobiotics come in. So they become very hydrophilic, it becomes water soluble. So immediately it will start coming out through the urine. Urine is one of the biggest places through which this comes out actually. So liver does that. Any molecule that comes inside, they do first, there are 2 stages of biotransformation. The first stage is they put in OH group or they put in oxidation. So the molecule becomes very hydrophilic. And then they also put in some other molecule. So it becomes very, once it becomes very, very hydrophilic, immediately it gets into water and it comes out through the urine. So urine is one of the biggest routes by which material gets eliminated from the body actually.
But why you told that it cannot come out of sweat? Like, sweat is also like water dissolved minerals which come out of.
Yes, but sweat, if you see, it's more hydrophobic in nature, the surface itself. So only hydrophobic materials can go inside. So when I put water, why water does not go inside my body? It does not go because it's very, very hydrophobic. Otherwise, if I stand under a shower, I could swell also, right? If the water goes in. It's not, because the surface is very hydrophobic actually. That's the main reason. So urine is one of the biggest thing through which salts get removed. So one of a fantastic, I would say, elimination process. So the idea of the liver is to oxidize your material so that also dissolves in urine and gets eliminated. So that's one. So any xenobiotics is any toxic material, any chemical gets biotransformed by the liver and then it gets eliminated through that. Fecus is another route. It's little more hydrophobic, comes through the fecus. Gases is very rare because the material has to get completely vaporized. So that means it should be only a small molecule. Big molecules will not get vaporized. But still, like if there is carbon dioxide, sulfur dioxide, nitrous oxide, all those things can come. But big molecules will not become a gas. Okay, so biomaterials, we have seen a lot of different types of materials. So they are classified like artificial, natural. Artificial, you have organic, like polymers. So many polymers. I talked about polylactic acid, polylactic glycolic acid, polyethylene glycol, polyester, PMMA, PEG. So many polymers, almost 20-30 polymers are there which are used in our body. If you go to metals, stainless steel, cobalt, chromium, titanium, nickel, all these are metals. If you go to inorganics, ceramics, phosphates, sulfates, oxides are all ceramic, copper oxide, nickel oxide, those so many things, they're all ceramic. Polymers, I talked about actually. Biopolymers, these are naturally occurring material, they are called biopolymers, okay, from say plants, glucons are from plants, from seaweeds, some polymers, carrageenan, agar, they are all from seaweeds. If you go to plants, you have glucons, you have xylons. From bacteria, curdlan, some sugar-based polymer. So you can have bacterial origin, you can have animal origin, you can have marine origin. Different materials are there. They are called biopolymers. The advantage is they will degrade, they are not toxic and so on, but they have limited supply. Suppose I want to say a material, a polymer from plant, then I have to have so many plants to get the enough material. So the availability is a big problem. That is one of the main problem, and they do not have so much strength like your polyethylene or other synthetic polymer. Okay, your synthetic polymers have a lot of strength, lot of life, all those things, but biopolymers do not have all those extra properties. So we are always mixing with biopolymers, with natural polymers to get the best suitable conditions actually. So there is a combination. Metals and polymers are used, polymers and biopolymers are used. We coat metals with biopolymers, we coat metals with ceramics, like that, different combinations. Like I've been talking about different parts of the body for different time durations, different environment in the body. So we need to design material in all these type of situations. So the one material does not work for all conditions. So there is a lot of research that's being done in medical biomaterial area actually. There are many companies globally. In India also there are many companies which have started working because it's a big area. Practically all of us have some outside biomaterial in our body, if not today, at some other point, like I said, contact lenses, stents, and urethral stents and shunts, or dental related material. So all of us, I think slowly will have some biomaterial in the body as time goes. So it's a very big research area for one to think about actually. It's a very exciting area because how the host, the body reacts to the material, how the material reacts to the body. So a lot of immunology comes into, a lot of molecular biology comes into, a lot of microbiology, because these biofilms are all related to microbiology. So all the areas of biotechnology combined with material design. So material scientist or inorganic chemist, polymer chemist. So it's a combination of many areas which come into. That's why it's a very exciting area to work on actually. Okay, any more questions anybody has?
To release hormones into the what?
Degrading polymer?
Release hormone instead of like drug. Like for people who need hormone therapy, like instead of taking pills daily.
That's correct. So if I am talking about targeted delivery, I want to target in one place. Suppose somebody has a liver cancer. So the drug has to go and bind to the liver, not to rest of the body. Suppose I just take the drug orally, it will go to all parts of the body, but I want it to the liver. We can have targeted delivery with the biodegradable polymer, which will go and then release at the place of liver. Somebody has a tumor in one particular place. So you want the drug, because most of the cancer drugs are very toxic. It kills good cells also. So we want the drug to go exactly to the tumor and then enter and then release the drug. So we need to think about that sort of targeted delivery with biodegradable polymer, how to target. So those challenges are there. What type of material which will go and bind to those tumors. So we need to think about that also. Yes. Any other question? Anybody has?
So they use mercury amalgams to fill in the teeth, right?
Yeah.
So won't that cause mercury poisoning?
Do what?
Won't that cause mercury poisoning?
create
Like
poison.
like mercury
there,
is
yeah. There are a lot
toxic,
of studies. The mercury does not get released. Even recently there are studies, but they are slowly trying to come out of mercury. Mercury was used because it's a soft material, so it can nicely fill up the holes. Because when you have a hole, the shape of the hole is very complex. So mercury amalgams can fill up. But many studies are done to see whether a mercury release is there. There is no mercury release, but
Can they use, like,
as I said, they are slowly trying to come out of that.
can they use any other like soft metal, like let's say gold or gallium or something?
Which one?
Gallium or gold. Gallium is also like melting.
Gallium chloride, is it? Again, see, we have to look at its softening temperature, because the doctor does not use any heating and to allow it to
Yeah,
spread
gallium
around.
is it's almost liquid at room temperature.
Equivalent. So again, it may have its own metal toxicities. One has to study those things actually. But metals, like I said, some patients might not have any toxic reaction for some metals, but same metal may have toxic reaction for some other patients. That is there. Nickel toxicity. Some people do not are able to accept nickel in the implant, but some patients do have. And nowadays, that sort of adverse reactions are increasing. Nickel, chromium, it's increasing nowadays actually. So there is a thought whether we can replace all metals with oxides, phosphates, carbonates, sulfates, like that, oxides, metal oxides, which are more adaptable in the human environment. So that's being thought of. But as of now, still nickels are there, chromiums are there, more of an amalgam. So it's not getting released, more of an amalgam. There are cobalt chromium amalgams, nickel chromium amalgams, and so on.
Can they use synthetics for that purpose, like just filling the gap?
Synthetic polymers are used, yes.
Okay.
Yeah, it is used. There are some adhesives which are used, yes. But the problem is how do they set at room temperature? Because when you are talking about dental, it has to be in room temperature. So there are synthetic polymers which are used, which will set at normal light, UV light. So if you have gone to a dentist, they will fill it up and then put a light. Anybody has seen that? Yeah, you have seen, right? They put. So originally they used UV based polymerization, so that the polymer will set when you have a UV. Nowadays, normal light based reactions also are happening actually. So originally they used to use heat based, but you can't do that in our mouth. So light based polymerization. So there are some polymers which cross link when you put in light.
So could they use biopolymers for the beads so that it degrades? Like, is there research being done using
there are already many biopolymers being used actually. Carrageenan based material, chitosan based. Chitosan comes from fish bones, they extract chitosan. Carrageenan comes from seaweeds. Then they use, there is one polyhydroxy, PHA, polyhydroxyalkanate, which comes from these chicken feathers, chicken head. There is one head is there, no? Chickens from there. So there is a lot of polymers. But the main problem is quantity. Suppose I want 100 kg. I have to go to fish market or how to get. So the limitations of availability is a problem with biopolymer. So it is good to go for bacterial based. Then I can do a fermentation and do that actually. So some biopolymers are available based on bacteria, glucon, sugar. Then you have curdlan, which is available from baker's yeast. So from baker's yeast is used in fermentation. Yeah, so they extract it, and we can use it for different applications on biotechnology. So animal based is a problem because animal availability, but if you can go into bacteria or yeast based, it's very good. We can get more. It is still used. You go to CMC Vellore, you have heard of CMC Vellore. Many hospitals use. They use PMMA still. Polymethyl methacrylate is cheaper and they are used to it. Yes. Like I said, biopolymer availability, quantity, all those still an issue. So you have to mix it with some non-biodegradable synthetic polymers. But if you look at cardiovascular stent, there is a stent, nickel titanium, then they'll coat it with a biopolymer with the drug inside. So the biopolymer will slowly degrade and the drug gets released. So they are called drug-eluting stents. So there is a bare metal stent, drug-eluting stent. So if any of your relatives had angioplasty, you can ask them what type of stents they have. Two types of stents are there: bare metal stent, there is no coating, it's cheaper, and then you have the drug-eluting stents where you have a biopolymer coated on the stents which will release the drug. They are more expensive, almost 2.5 lakhs, 3 lakhs. The bare metal is like 1.5 lakhs, like that actually.
Sir, is there any permanent solution for the biodegradable or degradable materials to bacteria settling? Because this is not fully permanent, right? Because it erodes fully, and again and again to get coating, it will be.
Okay, so this bacterial infection happens in the early days, first 7 days. I have a wound. Bacteria has entered. If you can somehow manage the 7 to 10 days, there won't be any bacteria inside. Normally there is no bacteria in our body. Only when there is a wound, open wound, accident, trauma, bacteria enters, and if you can kill for the first few days. And doctor does not give antibiotic forever and ever, right? He gives only 3 days, 5 days, 7 days, like that. So early days, once you can. But the other problems will be inflammation, reaction of the biomaterial to the body. Those are other problems. But the infection wise, 7 days is enough. You're okay after that.
Sir, how do they, like, if people are trying to try out new biomaterials for specific purposes, how do you test it on a human? Like, we can do all our calculations, but it might not always suit a human being.
So once it is tested in the lab, you have the preclinical trials where they do it on 2 animals. So you have preclinical, they will first do it on rats or mouse. Later on, they will test it out on. Later on, they will test it out on monkeys, or if it is a orthopedic, they will test it on even horse also. Then it comes to human. Okay, so 2 animals, then that is preclinical. Then the clinical trials come. So they will have phase 1, phase 2, 3. Clinical phase 1, phase 2, phase 3. So it will take around 4 to 5 years. So this may take about 1 year. This may take about 4 to 5 years. So totally about 6-7 years. All the tests are done. So, then you submit the.
Clinical means they will take in volunteers.
2 types of animals first they will test.
No, sir, clinical.
Clinical, human.
Volunteers.
Clinical 1, clinical 2, clinical 3.
Human volunteers will come on.
Yeah, human volunteers will come, and that will clinical test 1, 2, 3. So they will test it out later on the real patients who are willing to volunteer that particular material. So totally about 6 years it will take. So the toxicity is the first step. So on animals, when we do on rats, we do 30 days. We put that material inside the animal, rat, and then sew it, and then take it out after a month and look at toxicity, inflammatory response, and all that. That is the first thing. How it works comes later, but first is toxicity, whether it causes any reaction to that. And then they may test on horse. Suppose a horse with a broken leg, they will test it out. So that is second step. That is preclinical. Then they will go to clinical trials actually. So the second step is normally big animals. It could be dog, it could be sheep, it could be pigs, it could be horse. The first step of preclinical is rats or mouse or something. Then clinical is human. Clinical phase 1, phase 2, phase 3. Any other question?
Sir, sir, what do they do to ensure that while they're operating and putting in the biomaterial, like there's no other infection?
Okay, that's very important. They sterilize the material before it is put in.
Not materials, like when you open body and put the material.
Yeah, so the material first is your surgery room is sterilized. The material is also sterilized. There are different ways of sterilizing. Metals means they sterilize at a high temperature, 125 degrees at 5 bar pressure. So all bacteria. If it is non-metal, like polymer, you can't use high temperature because polymers will break. So they use ethanol or sometimes UV or they use gamma rays. So different sterilization methods are there. So all materials have to be sterilized before it is placed inside. So it's in a surgery, normal surgery, like the doctor does an operation, no? Somebody does an operation. But the material is also sterilized before it is used. Like your injection, you must have seen, no? Doctor removes that thing. So that's sterilized actually. So he removes that and then takes the needle and then does that. You know? Exactly, all the biomaterials are also sterilized. So that's also a big area because polymers you can't sterilize with steam. Polymers will swell and break it. Metals you can sterilize with steam. Polymers, sometimes UV can degrade polymers. So ethanol. So those are many challenges in what type of sterilization depending upon the material used also. I think we will stop with this.